Hello and welcome to the next in our series of Conversations with Giants in Medicine. I'm Ushma Neal, and today I'm delighted to be joined by Sir Mark Feldman of the Kennedy Institute of Rheumatology at Oxford University. Feldman's research over the last 30 years has focused on the understanding of autoimmune disease, specifically the treatment of rheumatoid arthritis. His hypothesis about cytokine inhibition via TNF blockade was considered heretical in the 80s, until clinical trials led by him and his colleagues showed that it could be the cornerstone for treatment of rheumatoid arthritis. Remicade, infliximab, and Enbrel, Etanercept, and others are now the cornerstone of a $14 billion industry in TNF-alpha blockade. I look forward to hearing a little bit more about the man behind the translational research. Can you start by telling us a little bit about where you grew up?
Well, I grew up mostly in Australia, but as an immigrant. I arrived in Australia when I was about 8 and a half from France. But that wasn't my first home. First home was I was born on the Russian-Polish border. My father was lucky enough to be able to take his family out of there at the end of the war and went to France, where he had a sister that helped to get the necessary formalities. But somehow life in France in the immediate post-war era was difficult, and so he wanted to move on. He had two cousins, both doctors, one in New Haven, one in Melbourne. They both filled out immigration papers for him, and the Australian ones came first, so off we went to Melbourne. And that's where I grew up. It's a wonderful place to grow up, lots of space, good weather, and a very good educational system.
Was your father a physician or a scientist?
No, he was an accountant. And despite the fact that he had trained in accountancy in France before the war, in the immediate post-war era, things were difficult and he wasn't allowed to practice as an accountant, and so it made moving on away from there to a land of more opportunity desirable.
Now, when you were a child, did you show an early interest in science and medicine? Where did your interest lie?
I don't think so. I think at school I was a diligent student. I was quite keen on doing well at school. I think the immigrant ethic is to work hard. That's the road to moving on, and so I was a good student, but I wasn't particularly interested in medicine. I was interested in science. At school you do physics, biology, chemistry, but nothing really about medicine. So it was just a way of moving on. But my father's cousin that had done the immigration papers for us was quite a charismatic and entertaining person, and he seemed to make medicine into a very glamorous career. And so both my brother and I decided that was actually more interesting than accountancy, and so we both went to medicine at the University of Melbourne.
So, according to your CV, you graduated the top of your class of 200 students, the best in 8 out of 14 subjects, so it was clearly something that when you got there you excelled at.
Yes, and I think it was still the immigrant ethic. If you're going to do something, you do it well. But I did find it really exciting and interesting, and so it wasn't an unpleasant task to do well. And the educational system in Melbourne was very good at the time. The teachers took, I think, more personal interest probably than they have time now. Now they've probably got to get their research papers or else they lose their positions. Then I think it was probably a slightly more relaxed era. So the teaching was good, the encouragement of students was good, and I really enjoyed my time at the University of Melbourne, met my wife there, got married while I was a student, so it was a good time.
Did you do any clinical practice before launching straight into your PhD, which you did with Gus Nossel at the Walter and Eliza Hall Institute?
Yes, when I was doing medicine, I didn't really know which direction I would end up. So the only thing that I was quite keen on was to learn clinical medicine effectively, and the University of Melbourne had two teaching hospitals. One, the Royal Melbourne, was very close to the medical school. The other one, St Vincent's, was about 2 miles away. One took about 80% of the students, one took 20%, so it doesn't require much mathematical insight if the hospitals are the same size to realise you'd see a lot more interesting patients in the teaching hospital with less students. So I picked that hospital, and it was really good for students. But the difficulty came later because as a student you just examine the patients you want to see. As a young doctor spending your full-time work in the hospital, you see all comers. And this was in the Irish part of Melbourne where people joke, but not that inaccurately, that there wasn't a pub on each intersection. There were 3 or 4. And half the patients had serious alcoholic problems. So I make a joke at times that I owe my research career to alcohol, which most people find quite impenetrable and invariably ask. But it was really because seeing a lot of alcoholic patients with problems that were not soluble really gave a focus that doing research to understand medicine in greater detail might be more interesting than seeing the same type of patients day after day.
Well then, given how many patients you would have seen with liver diseases, how did you stumble into doing your research with someone who was famous for antibody formation and immune tolerance?
When I visited the Walter and Eliza Hall Institute, which students never visited, they didn't have undergraduate students, I realized this was quite a different place. You could feel the atmosphere was different. It was sort of a very exciting place, and in about 30 minutes it became pretty clear that that's where I wanted to go. So I was very fortunate. I didn't know for many years that I applied far too late, that Gus Nossal had already chosen 4 PhD students. There were 5 units, but that year there was only 4 students because he was going off on a sabbatical to Paris. But he didn't tell me that, so he told me that if you want to come, you're on. So he had to get an extra fellowship and he had to find an interim supervisor, which he duly did. And so I turned up in February, totally unknown that I'd bypassed all the rules and that Gus had effectively shown some judgment to launch me on a potential research career.
Well, after what seemed like a very productive PhD, you then decamped back to Europe, going to England to do postdoctoral research at the Imperial Cancer Research Fund on tumor immunology. So why jump from the immune system in general to something as specific as tumor immunology?
Well, it actually wasn't very specific. I mean, the reason I moved to England was to work with Avrion Mitchison. While I was a PhD student, I had another piece of good luck, which was that the Hall Institute went into a financial crisis. In a financial crisis, the leaders have to bail out the ship, so Gus Nossal had to remain home, and so he sent me on a number of conferences where he had been invited, and they were wonderful because as a young student you got to meet many of the immunological stars of the time, and you could learn you could see who you might work with in a way that was quite different to usual. Usually with the problem in Australia is the tyranny of distance, that you're so far away, you feel out of contact, disconnected from the world. If you want to go anywhere, it takes you 24 hours. And so sending students to go and meet potential postdoc supervisors was never high up the agenda. But I was able to compare some of the friends of Gus that he was recommending, Jerry Edelman, Av Mitchison and others, and decide which one had the right personality that I wanted to work. And Av Mitchison, a very charismatic scientist to this very day, seemed to be a very interesting contrast to my Australian mentors. But the need for Australians to go abroad at the time was very well known. It was impossible to get a good job in Australia if you'd never left. And they were jokingly called a BTA or BTE degree. Been to America, been to Europe. But it wasn't funny, it was real. If you hadn't gone, and people would still remember you as you were in short pants growing up scientifically and wouldn't believe that you were ready for a proper appointment.
So you chose to work with Av Mitchison.
Yes.
On tumor immunology.
Not really. I mean, he had what was called a tumor immunology unit, but it was really, it was in University College, not in the main Imperial Cancer Research Fund campus on Lincoln's Inn. He'd inherited the chair from Peter Medawar in zoology, and it was really a general immunology department. And his approach to all postdocs, when they asked him what they wanted to do, was to say, well, you've got very good resources, what do you want to do? Just go and do whatever you want to do. And he was doing tumor immunology, and of course quite a number of people decided. I decided instead to continue at the time working on the cell interactions and to try and characterize some of the mediators that seem to be acting as signals from one cell to another. And that's what I continued to do for a while, and didn't really try and do tumor immunology for about 5 or 6 years. When I tried to do tumor immunology, I thought that the relevant tumor immunology as a medical graduate was humans. So we started to look at human cancer patients and found it was absolutely impossible to measure any immune responses. And so I made a conclusion which has been made before me many times and after me many times, that what you want to do for cancer is to develop an autoimmune disease. That's what you would like to have happen to cancer patients, get the immune system to target on the cancer. And so the next step, logically, was to ask, well, how do you induce an autoimmune response? And and since the Hall Institute had been the home of Macfarlane Burnet, where autoimmunity was first classically defined, this has been a topic I'd worked on a little bit during my PhD. So I felt that that was a reasonable next step to try and understand autoimmunity.
So you started working on autoimmune thyroiditis? Right?
Yes, well, I started to think how I might progress. And like everything in life, a little bit of good luck goes a long way. So this is back to about 1982, and a couple of papers had come out looking at the role of HLA antigens and their upregulation in sites of autoimmune diseases. And one of these groups was based in the neighbourhood about half a mile away at the Middlesex Hospital, where the stars were Ivan Reut and Deborah Doniak, but the most active and noisy person was an Italian called Franco Battazzo. And so he and Deborah came to visit me one day, and because one of my research topics in recent times had been cell interactions and how lymph T cells and macrophages and presenting cells interact, which involves HLA molecules, HLA class 2 particularly, and to ask what did I think this upregulated HLA class 2 in the diseased tissue site meant. And of course, if what you do the rest of the week is study HLA class II antigens in antigen presentation, when somebody shows you histology and you say, well, this is the visual picture you would expect if you had upregulated antigen presentation. And that was an hour or two's meeting. They disappeared. We said we would meet again. But I started to think and realized quite soon that this actually was a missing link, that you could develop an idea of how autoimmune diseases were triggered and were maintained through the upregulation of antigen presenting function. And so that's what eventuated some months later. I wrote this hypothetical paper which was published in The Lancet in '83, so about 30 years ago now, and certainly to my surprise it's still not an embarrassing read. Some things have changed. Regulatory cells have appeared. They used to be called suppressor cells, but that's probably the major difference. And of course the molecular characterization. There was, so it was a fortunate encounter that led me to be able to link one of the things I was very interested to understand autoimmunity for using it for cancer, to link it to my understanding of cell interactions and some of the molecules with the histological picture. This was a fortunate encounter, and while I was aware of these papers, I think talking to people made it so much easier.
What led you to starting your work in animal models, at looking at the cytokines in joints?
Yes, well, the consequence of this new idea about mechanisms of inducing autoimmunity was to test it, because having decided one had this idea and then deciding we may as well publish it without any data, the consequences of that act soon dawned, and that we better actually test it. And subconsciously, I think we probably would not have gone ahead and submitted the paper had we not had all the tools to test it, because we had T-cell clones. We were studying these cell interactions all the time. And so it only took about a year to really test the cellular basis of this idea. And so in a way, I think this is a prime example of the importance of a very clear testable hypothesis in driving science. Once autoimmunity and antigen presentation were linked, then many other things become automatic, that what upregulates antigen presenting molecules at the time, the only known signals were cytokines. And so this led the focus quite quickly to studying cytokines.
And I imagine this must have been part of what drove you to look at the joint as opposed to the thyroid, given the access to tissue that you could have.
Absolutely. But it was modulated. The reason we had been interested in thyroid, and the reason thyroid was the best studied human autoimmune diseases then, and actually to this day, is that medical therapy is very pro-scientific. By the time you need thyroid surgery, your thyroid is large, so 50 grams or more of material gets removed, very easy to put into a pot and have lots of cells for research. But the other important thing for science is funding. And I think, of course, these days you don't have any scientific conversation or any conversation with scientists for more than 10 minutes before this comes up. But it was always the case. So having a lot of money for studying thyroid disease in great details has always been difficult, because the current concept is that the therapy of thyroid disease is actually pretty good. it's not perfect, but it's good enough that there's no incentive to improve it. And so to be able to move on, we had to consider what other disease had many of the local features, had reasonable amounts of tissue, and could be studied. And so rheumatoid arthritis seemed to be a logical consequence that could be studied with the same technology.
And not to mention that it happens to approximately 1% of the worldwide population, and so therefore would be a bigger market to study.
It would be much easier to get grants, and in fact, from the moment I expressed an interest, collaborations, that's when I met my long-term colleague, Ravinder, better known as Tiny Maney, and we've worked together now more than 25. Well, we started in '84, so it'll be getting close to 30 years. But he is a scientifically educated rheumatologist, and had been also interested in mediators from cells during his lab phase, and so it became very easy and very enjoyable to work together. And of course, there was a lot more money for research.
So at that time, when you first started collaborating with Professor Maney, you were working in animal models, correct? And isolated human tissue to try and explore what cytokines were being produced by the tissue.
Yes, we started, we first had a little bit of an abortive attempt, because before we started working together, the biggest rheumatological problem was systemic lupus. It still is. It's a less common disease, but its downward course is much faster. And so both diseases were being looked at by Tiny Maney. And because the autoantigens in systemic lupus were starting to become defined, there were nuclear antigens, we thought that might be tractable. The problem was that there's no local site of the disease, and using cells from the blood gave very consistent and entirely negative results. No immune responses could be measured. So after probably not much more than a month, we decided that that project wasn't going to work. Rheumatoid arthritis could work. And so it went in two directions. One was to start to obtain as much human disease tissue as possible, and then on the assumption that what we really had not learned from the thyroid was what were the important rate-limiting cytokines, focused on the cytokine assays. And there, of course, other lucky events took place, because having worked on these intercellular mediators since my PhD days, as the field developed, one was going to these conferences, one was taking part, you knew everybody. And so as cytokines progressively became molecularly defined, not by the immunologists, but by the intervention of molecular biologists, these tools became available. And these tools were used by us on human disease tissue ahead of the great majority of people that might have done so. So one of these strokes of luck was starting to fish in the cytokine pond before others knew that it might be very important for that type of disease. I think people were very interested in cytokines as cancer therapeutics and not as drivers of autoimmune diseases. That was not a popular concept.
According to what I remember, at this point you had sort of hit on IL-1 as being one of the main inflammatory mediators and went through a series of processes of trying to figure out how to neutralize that. And you hit on TNF-alpha.
Yes, that, well, it wasn't that we thought our interleukin-1 was that important for the disease, because we were doing the research, so we were quite agnostic. We were actually looking at everything. But the consensus view, which was based on what cytokine injected into a joint of a rabbit gives you the biggest inflammation, and that was interleukin-1. So if you were not not working in that field, you thought the answer's obvious. So there was quite a lot of thought, and actually, the real dilemma we faced as we catalogued inflammatory mediators and other cytokines in joints is that there was a very large number of mediators expressed. So naively, as the cytokine field grew, we started analysing what was in the joints when there may have been 6 to 8 molecular defined cytokines. And so the initial assumption was that what's going to be present will give you a clue to what's important. But we didn't realise to what extent our molecular biological colleagues were diligent, would produce more and more molecular clone cytokines, and that if we used the tools, we would find that every known cytokine was present in the rheumatoid joint. And that was quite a paradoxical result, because in a normal situation, if you stimulate a blood culture with PHA or give a mouse endotoxin, nearly any cytokine you want to measure, interleukin-1, TNF, interferon gamma, will be produced for a day or so, and then it switches off. So if you look at cytokines in diseased tissue and everything you can measure is upregulated and present, it suggests that the normal short kinetics of production has been deregulated. These things are produced over a long period of time. So if you take a snapshot, they're all there because they're produced. And that was a real paradox, but in a way was very, very important, because it told us that our generic assumption was correct. The cytokine system was deregulated in a major way, and therefore focusing on that to work out which may be the rate-limiting step would be likely to be productive. And in that context, it doesn't matter where you start. As soon as you start the experiments, then the results would guide the progress. So there was no need to not look for what regulated interleukin-1. We were not convinced that that was the wrong answer. We just wanted to know, what regulated it, because the therapeutic dilemma of the time was quite obvious that if you had, let's say, half a dozen pro-inflammatory cytokines whose properties overlap considerably, you couldn't block them all. You would have to block one. And then the question was, which one? And of course, most people thought that if you've got many cytokines, overlapping effects, what was called redundancy, then blocking a single one wasn't going to work. And so many of our competitors at the time, there was probably half a dozen other groups interested in this field, decided that cytokines were not good therapeutic targets.
And yet you persisted and hit upon using TNF-alpha blockade.
Yes, we persisted basically because the properties of cytokines seem to be really important. I mean, things which are very potent work in very small concentrations and set up cascades of interactions of cells and of biochemistry, suggests that they're important. So perhaps knowing a little bit more about the biology of cytokines, having grown up with the field, made it easier to be quite convinced that this was an important source of targets. And of course, now today we know that most cytokines can be blocked and develop a therapeutic benefit in different diseases. So the most abundant source of therapeutic targets we know is in fact cytokines and their receptors. But we persisted because we just couldn't believe that these very potent molecules present in such abnormal quantities, in abnormal kinetics, were just going to be a byproduct and weren't really a major driver. And so I was very fortunate that my PhD project enabled me to, with some very talented postdocs, Glenn Buchan and Fionnuala Brennan, to work out how to culture the synovium so that all the cells survived. Because to an immunologist, one of the most amazing aspects of cell culture of joints was that the rheumatologist took a mixture of cells that were 90% immune inflammatory, 10% fibroblasts, put in a dish, and after 2 days threw out all the supernatant cells, everything immunologists would think interesting, and keep the adherent fibroblasts. So to an immunologist, this was the world's most surprisingly bizarre experiment, and how people thought that this reflected the biology of arthritis is interesting. But it's like certain things, they get used, they get repeated, and eventually people don't realize that it doesn't reflect what was happening in vivo. So we were able to develop these methods, and then Fionnuala Brennan was able to do what was the pivotal experiment, which is to put in antibodies to TNF into a mixed synovial culture and show that interleukin-1 and then lots of other inflammatory cytokines were downregulated.
So this quickly took off, such that in 1992, you and Tiny Maney started your first open-label, non-blinded trial in 20 RA patients that were otherwise refractory to other treatments at the time. Can you tell me a little bit about how that went and your excitement at the time as the data was coming back?
Well, the excitement was very palpable, but there were a few little hurdles, one of which was that we had developed this idea based on the blockade, the TNF blockade downregulated other cytokines, that it was the therapeutic target. But everybody and his brother knew that autoimmune diseases were driven by CD4 T cells, and so the companies that were starting to work with monoclonals were very busily trying to cure rheumatoid arthritis with anti-CD4 antibodies. So as we started to try and get some traction on our idea, we quickly found that they weren't interested.
But in this case, your former student, Jim Woody, was at Centocor.
Yes, that was very, very fortunate, although Centocor was also preoccupied with antibodies to CD4. And so Jim Woody was very, very pivotal, because the rheumatologists and the clinicians at Centocor were doing anti-CD4. But he was senior enough that he was able to, by himself and a few helpers, help us do anti-TNF. And so, it's interesting to reflect what may have happened under different circumstances. And one of them is what happens if your pet project is given to a student or a postdoc that isn't very good. What happens if they give you the wrong answer?
And yet you had stunning success. So did you hear from your patients fairly quickly that this was finally working for them?
We thought our data was very convincing, and so we wanted to do a formal clinical trial right from the beginning, with placebo controls. But it turned out that the perception that blocking a host defense molecule, TNF, was dangerous was too ingrained to enable us to go straight to placebo control. We had to do an open study first. We could not convince anybody that since anti-TNF had to this time been used in several thousand patients that have severe infection, that have bacterial sepsis, testing Tony Sarami's idea that sepsis was driven by TNF. So if it was okay for patients who already had the infection, why couldn't we give it to people without infection? We couldn't convince anybody that this was a huge, bizarre misinterpretation of risk, but we had to go along and do an open study. It took about 3 hours for us to get a clue that things were going right, because as we gave infusion of infliximab, this is the current name, but at the time it was called CA2. The nomenclature is a funny one. C stands for chimeric. The antibody was sort of 3/4 human. The A2 was the code number of a well where the right hybridoma appeared. This came out of Jan Wilczek's laboratory at NYU. So CA2 was the nomenclature at the time. It was given slowly to avoid infusion reactions, but as the patients were getting this, the first patients, they told us they were starting to feel much better, less tired, and so we had a little bit of a clue within a few hours that something was happening.
Now, given that it was a chimeric antibody, did you ever have concerns that if you had to give a second dose, which it did show up later on that these first patients, the effect wore off after 6 to 12 weeks, did you have a concern that it was going to be immunogenic?
So we were reasonably optimistic, and so we decided to ask for ethical permission. By this I mean me. I'm an immunologist. Tiny Maney is the clinical rheumatologist. So really he was the one that was doing most of this, and I was encouraging him, and between us we sort of reassured each other that this was all safe, rational and ready to go. But he got permission from the ethical committee to retreat these patients as they relapsed, because while the first experiment was a relative success, 12 weeks is 3 months, that's a long effect for a short course of injections, but in the lifetime of a patient, it's not enough. So it needed more therapy. And the miracle we might have dreamt about, that we might have cured these people, certainly hadn't happened. So we needed to carry on, retreat. And so that was done on these patients from this open study.
But the further trials that you did after that, combining it with methotrexate and spacing everything out, and then later on the trials with etanercept, which was a TNF trap, and Humira, which is fully human, have now led to the gold standard, such that you can not only treat symptoms, but stall the destruction in the joint where it starts when you start the treatment.
No, that's right. So the challenge we had, at this time, this was about we started in May 92. So in a few weeks, we knew things had worked. Round one, that we had established that TNF was an important molecule. And perhaps a testament to how interested our sponsor company, Centocor, was, is that the results were disclosed in September 92. So before they were published, they were announced. One of the things that I like doing, or I've grown to like, is organizing scientific conferences, because communication is a very big rate-limiting step in probably all of life, but especially in science. And so we'd started organizing translational research conferences from '84. So in '92, we had one in Israel on the Dead Sea, and we asked the Centocor if we can disclose this data, and no problems. Despite the fact people from Roche, from Amgen, from Immunex were all coming. And so, surreptitiously, this decision to disclose was actually very useful for the community and the patients and the progress of medicine, because it set up competition. Roche and Immunex and had already made TNF inhibitory molecules for use in sepsis. Sepsis wasn't working. They were told that rheumatoid worked, so they started working on that. And I think the competition is always very good for the patients. And so maybe that's actually how science should work, that academics should be defining, targets not only in the laboratory, but doing the first proof of principle. And then the data can be used by many other companies. Unfortunately, that's not usually how it works. So.
Well, at this point, as I mentioned in the introduction, TNF inhibitors have been used in ankylosing spondylitis, in Crohn's disease, ulcerative colitis, and other autoimmune disease, and it's now a $14 billion industry.
No, I have to correct you. It's 25 billion.
25 billion.
Yeah.
I'm sorry, I underestimated.
No, well, it's hard to know the figures, but because the databases are kept by companies that only let you have them for large sums of money. But I only found out it was 25 billion a year ago when I began to give a lecture at Merck, and the introduction was that, here is one of the people that discovered TNF as a therapeutic target, and TNF has now surpassed statins as the biggest drug class.
In the past, you've written that you did your science, you did these clinical trials because you were interested in the science, curious, you wanted to treat patients, but in the end, you've also been lauded with several honors and awards, including the 2003 Lasker Prize, and in 2010, you were knighted by Queen Elizabeth II. What was that process like? Do you have a sword? Did you get a cape? Did they give you a white horse as well?
You don't get it. They don't give you anything, but in fact, it's quite funny. I mean, in my the Queen is now mid-80s, so now it's nearly always done by Prince Charles. And before the ceremony, they sort of write to you and ask some interesting questions. And one is, can you kneel? Okay. And then, can you get up? These are interesting questions, but if you look at the average age of people getting knighthoods, it's not entirely. But it was a very nice ceremony at Buckingham Palace. There were, they give probably about 24 knighthoods a year, and so it's broken up. So there's the day we were there in October, I think 2010, there was 3 knights. One was a gentleman who had been the British general in Afghanistan. By the time he got his knighthood, he was no longer the British general in Afghanistan, because he thought that a general should show leadership, tell the politicians what's going on, and basically he was saying that no Western country has made any progress fighting Afghanistan for 150 years. We certainly won't. Why are we killing our young men? Well, that's a recipe for moving on. They still gave him his knighthood. And the other third one was a gentleman from New Guinea who got his knighthood for services to Papua New Guinea football, to rugby league football. And I thought that was a very interesting group. But probably the awards in society, including science, is useful as a motivational, aspirational tool. And I can tell you it is nice to know that you haven't wasted all your time doing research, and you can convince your parents, relatives, spouse that yes, you haven't been wasting all your time doing this work.
If you weren't wasting your time doing all this work, what vocation do you think you would have chosen otherwise?
Well, I grew up in Australia, so this is a sports-mad culture. So I did play a lot of tennis, and I enjoyed it. Tennis was a very low-ranking sport in the social hierarchy in Australia. In Melbourne, it's Australian rules football, but I'm about 1 foot too small for being good at Australian rules. So instead I was playing tennis at school. But that had an advantage because I'm 100% certain that none of my school friends are still playing Australian rules more than 50 years later, but I can still play tennis, and I enjoy playing tennis with my son. But I wouldn't have never become a professional tennis player. I think they also are rather more physically substantial specimens.
Well, thank you so much for joining us today. I really enjoyed speaking with you.
Yeah, it's been a pleasure to come.